I am Sarah Cryer. I am a postdoc at the Lyell Centre at Heriot-Watt in Edinburgh. And today I'm going to be talking about a part of a project known as CHALKY, which is looking at coccolithophore controls on ocean alkalinity. So CHALKY is a £2.2 million project led by Professor Alex Poulton here at the Lyell, but it's a multi-institutional, multi-country project with many partners across the globe. And its main, the overarching question of CHALKY is, how do coccolithophores affect the potential for seawater to absorb CO2, and how will this change in a warming ocean? But if we take that back a step, so we just think about how these two things relate. So the ocean has taken up about 30% of anthropogenic CO2 emissions, but we've only had a decrease in pH of about 0.1 units. But as we continue through the rest of the century at current emissions, we're going to see a further decrease of 0.2, 0.3 units. And so I'm just going to explain why we've only had this small decrease since the pre-industrial time till now, and how this influenced by how this might relate to coccolithophores. So ocean pH is controlled by the concentration of hydrogen ions, and it's on a negative log scale, which means if there's more hydrogen ions, it's more acidic, so lower pH. So atmospheric CO2 dissolves into the surface ocean, and this is then forms carbonic acid, which can dissociate into bicarbonate ion and carbonate ion. And these different species are what forms dissolved inorganic carbon, which makes up the largest pool of carbon outside of the lithosphere. So it's a hugely important source of carbon and store of carbon. And so if we were just to think really simply, what would happen? You might think, okay, as CO2 increases, we're going to have an increase of hydrogen ions, which is going to decrease the pH. But this doesn't quite happen. And this is because of the carbonate system's ability to buffer the addition of CO2. So these bicarbonate ions and carbonate ions will mop up excess hydrogen ions. The reaction will go in reverse, and we will have pH, or the concentration of hydrogen ions, can try and remain constant, and pH can remain stable. And it is these carbonate ions and bicarbonate ions that we think of as alkalinity. So the ability to mop up hydrogen ions, and there's a few other negatively charged ions as well. And so therefore we could think of ocean alkalinity as the ability of the ocean to absorb CO2. So that's kind of our alkalinity side of things. And then if we think about coccolithophores, so coccolithophores are single cell phytoplankton that produce calcium carbonate plates. So this is an example of all different species of coccolithophores. So we have larger ones here, we have slightly smaller ones here. This one that I'm pointing to, I hope you can see my mouse, this large one here is the same species as this, it's just a different life form, but they form all these different interesting calcium carbonate plates. And this is my favorite one here. And this is what we think of as the coccosphere. So it's where all these plates are forming together, and then they're made up of coccoliths. So you might hear me refer to them as liths, and these can be shed as well and be in the water column. And you can see in all these images, there's quite a lot of loose liths just hanging around. And the most common or the most studied coccolithophore is Gephrocapsa huxleyi. I'm not a biologist, so I am learning how to pronounce these Latin names. And it was formerly called Imelania huxleyi, and I'm, for the rest of the talk, going to call it EHUX or GHUX, so I do not have to say these Latin names again. So this is the most studied one, but there's also many other species present. And so coccolithophores photosynthesize and calcify. So you can see that they're going to have an influence on the carbonate chemistry, because they can take up CO2 and through photosynthesis and convert it to organic carbon. But they will also take up bicarbonate ions and release hydrogen ions when they're calcifying to produce calcium carbonate. They will also release alkalinity through dissolution. So if the whole coccosphere can start to dissolve, or the plates can be dissolved, and this is going to release carbonate ions back into the water column. And therefore, how coccolithophores live and die influences the fate of alkalinity, which is what the CHALKY project is looking into. So if we have the production of coccolithophores happening in the surface ocean, but we can also have grazing by microzooplankton and zooplankton, as well as viral lyses. So this is when the coccolithophore is infected with a virus and it can explode. And if all of this is happening in the surface water, then this is going to retain the alkalinity within the surface water. But the coccolithophores or the fecal pellets can be exported to depth, so this will have alkalinity loss from the surface ocean. And therefore, different loss processes are going to lead to different fates of the associated alkalinity. And it is the balances of these processes that are going to determine how coccolithophores control ocean alkalinity. And so my part of the project is looking at the production, the production side of things. So I'm interested in seeing what is controlling the production of the coccolithophores. So the research is focused on the Icelandic Basin. Sorry, this might be a little bit small, but I just want to show you where the Icelandic Basin is. So this is Iceland here, the UK would be over down here. And so it's this area of deep water here. And there were crews that happened last spring to try and capture the spring bloom of the coccolithophores. So there's two kind of sets of stations. We have 4 stations that create a time series where they were following a float, so it can be thought of as the same parcel of water, and then 5 roaming stations that were trying to find the coccolithophore bloom. And so we can measure the particulate inorganic carbon of the water column, and this will be all living and dead coccoliths and loseliths. So when I'm measuring PIC, I'm looking at everything that would be in this picture that is formed of calcium carbonate. So we have the pelagicus species here, the E-hux, G-hux here, and all of these loseliths. And it's not going to differentiate between living and dead. And when we look at the time series data, so we're going through these 3 S stations and then an R station, you can see that the PIC increases at all depths from 0 to 40 meters across through time. And this coincides with an increase in calcite production by coccolithophores. So this is total calcite production. You can see calcite production increases, and then starts to slow. So it kind of suggests that we have this increase in cell numbers peaking around this station 4 and then starts to decrease again. And there was 2 main species present. So when we look at our SEM images, the 2 main species are the coccolith pelagicus, which is a large species, and the G-hux, this one here. And it's the G-hux that sheds a lot of these liths. And we can look at the different cell counts. So this is using a flow cytometer, which excites the cell. It releases, it fluoresces, and we're able to, based on the particle size, estimate how many cells there are from the 2 different species. So initially I have an increase in pelagicus, and then this is followed by an increase in G-hux, which is shown here. And we can see, so this pelagicus, I'm sorry, I forgot to put this on here. This pelagicus is 100, it's been times up by 100 times to see, so that we can plot them on the same scale. Because pelagicus is a much larger species, it's going to have a larger calcite concentration. But when we're thinking about the alkalinity and the calcite production, we want to integrate this over the euphotic zone. So I've got 2 previous studies compared with our preliminary data. So this is Icelandic Basin study from 1991, and then a bloom study in the Patagonia shelf. And you can see that the calcite production, when we integrate across the euphotic zone, is much larger in the Icelandic Basin. And this is despite the fact that the standing stocks of calcium carbonate are comparable to the other 2, which this has very high calcite production despite similar calcium carbonate, which implies very rapid turnover and active calcification, which we're seeing in our calcite production numbers. And so then we can look at growth rates, and our growth rate is much higher than the Icelandic Basin study from the early 90s. And our turnover, we have much quicker turnover. So we're turning over that calcite at a much higher rate than in previous studies. So looking at how this PIC production and cell growth changes over time. So along the x-axis, we have cell growth, and then we have PIC production up the y-axis, and divided it between the two species. So the circles being GHUX and the squares being Pelagicus. And so we start up here. It's close to a 1-to-1 line at the initially. The growth rate then starts to decrease, but we still have high production of calcite, and then towards the end of the bloom, we have this growth rate and cell PIC production going closer back to the 1-to-1, but there is a difference between the species. And you can also look at this in terms of the cell calcite growth. So the figure on the left is just cell numbers. We can see cell calcite growth. And this time I've colored this by the percentage of calcite being pelagicus. So the dashed line is the 1-to-1, which is what we would expect if all the calcite produced is staying in the cells within the coccosphere. And we can see that when it's high proportion of pelagicus, so the darker purple color, it follows a more 1-to-1 line. And when we have lower pelagicus, so these like lighter orange colors, when EHUX is dominating, there is much higher, it falls away from this 1-to-1 line. And this is likely because the EHUX is producing, it's growing, and it keeps shedding these liths. So it keeps putting this liths into the water column. And this is just the image again to show you. So this here is the EHUX, and it's likely that a lot of these liths have come off this cell and it's producing, and it will also can start to produce multi-layers. So these are all images quite nice, nicely controlled liths where it's all quite close together, but you can start to see layers built up around the coccosphere. So we want to know, well, what stops the growth? So we have this increase in growth happening. As the growth increases, it then decreases again. What causes this? So nitrogen can often limit growth of phytoplankton, but we can see here, so I'm hoping you can see the bottom line because my zoom keeps covering it, but there are orders of magnitude different between the nitrogen required to support this growth and the nitrogen measured. So just looking at the time series, which is the triangles, we do have this drawdown of nitrogen going from yellow to orange to red to dark red, but we've still got plenty of nitrogen to support the growth rates that we're measuring. And the same can be seen for phosphate. So we have phosphate demand and measured phosphate, and again, we have plenty of phosphate to support this growth. And then finally, we look at iron. So we're still waiting for the iron to be analysed from these cruises, but we have this drawdown of iron. And these horizontal lines here I've plotted are just a range that has been measured in a previous study in the Icelandic Basin. So on a stationary point, if we just, looks like there's plenty of iron, but if there's that constant drawdown of iron, it might be by the time we get to the end of the time series and the iron demand is up here, that there may not be enough iron present to grow. And this is also assuming that nothing else is using the iron, but there is going to be diatoms also wanting to consume that iron. And when we just do a quick back of the envelope calculation, the nitrate and nitrite in the euphotic zone is 18 to 1,870 times greater than required to support growth rates. So you can see there's plenty of nitrate available, and phosphate in the euphotic zone is 4 to 450 times greater than required to support the growth rates. So this is suggesting that it's not the NRP limiting growth. And so just to summarize this is very much still work in progress. The coccolithophores are increasing the particulate inorganic carbon in the Icelandic Basin, but it's not the nitrogen and phosphorus that are limiting growth. So what is limiting growth? Is it the iron? Is it viruses? Is it grazing? And then the next questions is, how does this impact alkalinity? So I don't know yet. I'm hoping to find out. If you have any questions, yeah, I'm happy to take them.